Hi guys, today we're talking about DNA replication. So we know what DNA is and we know what its structure is. So let's talk about how it replicates to make more of it in order to make healthy organisms. Many proteins work together in order to help DNA replicate itself so cell division can happen and for DNA repair to happen. The relationship between structure and function is totally manifested in the double helix. Watson and Crick realized very quickly once they made their model that specific base pairing suggests to a possible copying mechanism for genetic material while allowing one strand to act as a template for your new strand. Base pairing is done based off of the template strand. Since the two strands of DNA are complementary to one another, each strand can act as a template for building a new strand in replication. That process leads to two identical DNA strands. The parent molecule, or the original strand of DNA, unwinds, and the two new daughter strands are built based off of the base pairing rules first identified by Erwin Chargaff. So here we have our parent molecule in dark blue. The strands separate, and the two daughter strands you can tell that are still identical because of our base pairing rules. The semi-conservative model presented by Watson and Crick of replication predicts what the double helix replicates. Each daughter molecule will have one old strand and one new strand. So the old strand is that gets conserved, and then a new strand attached to it, which gave us that semi-conservation model. There were a few other models that were competing with the semi-conservative model. The first was the conservative model, which said that the two original strands of DNA rejoined, and then you had one daughter strand that was entirely new. And you had the dispersive model, which was a mix of old and new strand. So here we see the conservation model, which over time would lead to too much new DNA, not enough original, like in the semi-conservative. And then in the dispersive here, it was kind of random old and new mixture. There were experiments that were performed that supported the semi-conservative model. They labeled nucleotides of the old strand to watch what happened over a few replications. And they realized that the new nucleotides were labeled with a lighter isotope, meaning that there was that semi-conservative model. The first replication produced a band of hybrid DNA, eliminating the conservation model pretty much right off the bat. That second replication produced both light and hybrid DNA, eliminating the dispersive model, which left behind only the semi-conservative model of DNA reproduction. This was their experiment. Some had heavy isotopes, some had lighter ones, replicated it, and found that the semi-conservative model was the only one to support their data. The copying of DNA is a remarkable process in and of itself, not only because of its speed, but also its accuracy. There are more than a dozen enzymes that work together, as well as other proteins to help replicate DNA. First, we have to understand, where does this start? DNA does not replicate from end of the chromosome to end of the chromosome. It replicates at regions called the origins of replication. And these are areas where the two DNA strands get separated, forming a bit of a bubble, which tells our first enzyme it's time to start replication and where to start replication. Eukaryotic chromosomes have many, as much as hundreds per chromosome, origins of replication, while prokaryotic chromosomes tend to only have one, sometimes two per chromosome. Replication does happen in both. So DNA replicates in both directions at the same time. This is how it would happen on an E. coli. We get our replication bubble forming here, It replicates out and then eventually pinches off. But in eukaryotic DNA, we have several of these bubbles that form, and then they connect to create the two DNA molecules at the end. The replication fork is where the origin of replication is. It's a Y-shaped region where the DNA strands become elongated. Helicase is one of the many enzymes that works together with all the other enzymes to help the process of DNA replication actually happen. Helicases are those that untwist the DNA's double helix at the replication fork and breaking the double bond between the strands. Single-stranded binding proteins then come in to bind to stabilize that single-stranded DNA so it doesn't reform its hydrogen bond. And our last one, topoisomerase, I always struggle with that one, corrects the overwinding ahead of the replication fork by breaking, swiveling, and then rejoining the DNA strand. Basically untangles the whole thing. So here, our unwinder helicase helps basically unzip the DNA. Think of this as like your zipper. Single-stranded binding proteins come in on both sides, and then primase is going to come in and prime the strand with a series of RNA to help prepare for replication. DNA polymerase is then added after the primer has been added. However, DNA polymerase can only be added to the 3' end of DNA. That means one of the strands is going to be replicated backwards while the other one is being replicated forwards. The enzymes called primase use that RNA chain from scratch to add the RNA molecules to the DNA at the starting region. The primer is very short, typically 5 to 10 nucleotides long, and the 3' end serves as the starting point for DNA. DNA polymerase is a series of enzymes that work together as one unit that catalyze the elongation of DNA at the replication fork. Most of the DNA polymerases require a primer in order to start, and they can elongate at a rate of about 500 nucleotides per second in bacterium and about 50 nucleotides per second per area of origin per replication fork in every human cell. Each nucleotide that's added to the growing DNA strand is a nucleoside triphosphate. This is DATP. It's a form of energy that supplies adenine to DNA that is similar to how ATP functions in metabolism. The difference is the sugar, where DATP is a deoxyribose, but ATP only has a ribose sugar. As each monomer of DATP joins the DNA strand, it loses 2 phosphate groups after it's passed its energy over. So here we can only build on our template from the 3' down. Okay, so we got our primer that comes in, and then DNA polymerase comes in and attaches the new nucleotides. This is an anti-parallel elongation, meaning it goes in both directions at the same time. The DNA polymerases can only add free-floating nucleotides to the 3' end, but we have one strand that goes 3' to 5' and the other goes 5' to 3'. The 3' to 5' we call the leading strand. That's because this can replicate continuously without interruption. The DNA strand that's elongated is in the 5' to 3' strand. That's the new strand, but we're reading 3 to 5, building 5 to 3. So that leading strand moves continuously through the replication fork. So here we have our 5' or our 3' to 3'. So this is our leading strand up top. Our leading strand adds the primer, DNA polymerase, specifically the DNA polymerase 3 comes in and elongates the leading strand. The other strand is called a lagging strand. This is where DNA polymerase must work in the direction working away from the replication fork. This lagging strand is synthesized in a series of short segments called Okazaki fragments that were named after Okazaki, the scientist who discovered them. They then later get joined together by DNA ligase, which acts as a glue to hold the backbone of DNA together. So here, this is how Okazaki described the Okazaki fragments, or the lagging strand, to be produced. So we have a template that comes down, DNA polymerase jumps in. Now we're working backwards. So DNA polymerase makes a primer. It can only go so long because this DNA polymerase is attached to the DNA polymerase on the leading strand. It gets pulled backwards, a new primer gets laid down, polymerase comes in and adds more. A few nucleotides before we hit the other primer, it's pulled back again, and then DNA ligase comes in. It removes the primer and then glues the backbone together, even though the overall direction of replication is going towards this side. If we're looking at this all together, we have our leading strand, which is nice and simple, goes on, but then we have our Okazaki fragments down here. Primer comes in, DNA polymerase comes in, it jumps back. And ahead, we have DNA polymerase 1 removing the RNA primer and ligase sealing the gaps. So this polymerase here is just going to keep jumping backwards to keep up with the primer and the DNA polymerase on the leading strand. Just some more ways of looking at it up close. This is a whole replication complex. So proteins that participate in DNA replication form a very large complex called the replication machine. DNA replication machines may be stationary during the process. There is a thought process that the DNA is moving, not the enzymes. Recent studies support a model in which DNA polymerase reels in the parental DNA and then gets rid of the newly made DNA molecules at the end. So this is what scientists are thinking has happened. Here's our parental DNA. It comes in, leading strand is being made, all the way down to our lagging strand, but DNA is really just being pulled through it, and our polymerases are doing the work, but DNA is what's moving. There's also a proofreading and a repairing function of DNA. DNA polymerase acts as a proofreader and repairer, as if it makes an error during DNA reproduction, it fixes it relatively quickly. In mismatched pair of DNA, a repair enzyme corrects the error in the base pairing. This means if like a C and an A bound together accidentally, our mismatched pair and DNA polymerase would come in, get rid of one of them, and fix it with the right one. DNA can be damaged due to an exposure to harmful chemicals or physical agents such as cigarette smoke or X-rays, even sunlight, and it can undergo spontaneous change or mutation. We have nucleotide excision repair, where a nuclease cuts out the missing or damaged pieces and replaces them with healthy pieces. So this is what nuclease does. We have this damage here, comes in, it'll remove one or two before, sometimes a few, and a few after, and the broken ones, repairs it, and then finishes it all off again. There is an evolutionary significance to this style of altered DNA nucleotides. There has been a rate or an error rate after proofreading that is very low. Typically, it's around 1 for every 1 million nucleotides added, which is a very, very low error rate, but it's not zero. Sequence changes may become permanent over time if they are passed to the next generation before they are repaired by the DNA. These changes are known as mutations, and they are the source of genetic variation upon natural selection operates. Blonde hair was actually said to be a mutation of the brown-haired gene. Same with blue eyes. That stuck around in the generation over time. When we get to the end of the DNA molecule, we have a lot of limitations of how DNA polymerase creates problems of linear DNA. The usual replication machinery provides no way of truly completing the end of the 5' strand of DNA. So repeated rounds of replication often shorten the DNA over and over again. This is not a problem for prokaryotes since they have circular DNA, but it is a problem for us that have linear DNA. So each time we lose a section of DNA off of the parental strand, which slowly shortens our DNA over time, which is actually how aging can happen. But eukaryotic chromosomes have something called telomeres on the end. These do not prevent the shortening of DNA because they are non-coding regions. They are just segments of DNA that don't mean anything to us. It prevents us from losing genes that are close to the end of our chromosome. It has been proposed to that the shortening of our telomeres is what's actually directly connected to aging. We do have an enzyme called telomerase, which catalyzes and lengthens the telomeres in germ cells to prevent those from passing on shortened telomeres. The shortening of telomeres might protect cells from cancer growth by limiting the number of cell divisions that can happen. There is evidence of telomerase activity in cancer cells, which allows cancer cells to persist and continue to divide.